So here we are again, picking back up with ribosomes, reminding you that ribosomes can be found on the surface of the ER or free-floating in the cytoplasm. The endoplasmic reticulum, or ER for short, is the first part of a larger organelle system called the endomembrane system. The ER is really a huge part of the volume of each cell, especially the ER combined with the Golgi apparatus, the two main pieces of the endomembrane system. They're a huge part of the volume because it is a system that is shuttling important proteins. The instructions come from the nucleus, they're made at the ER, and they shuttle things all the way from the nucleus through the Golgi to the plasma membrane. So it's a network that is allowing the nucleus to create or to code for a gene, transcribe a gene, that's going to code for an enzyme that may end up sending a signal to the rest of the body, but it all starts at the center of a single cell. So I want you to picture the ER as a bunch of interconnected flattened sacs and tubes. Much like the tunnels in an anthill, all of these passageways and open spaces inside the flattened sacs and tubes are interconnected, and the interior space is called the lumen of the ER. That lumen, or that space, is chemically separate from the cytoplasm, so it can be slightly different in, its electrochemistry and its pH. All of the folds of the ER allow for a great amount of surface area, and then the surface of the rough ER is called rough because of the abundant ribosomes that are dotting its surface, and they are synthesizing proteins while they temporarily dock on the surface. So, before we go any further, here is that endomembrane system, and you'll see this picture again. So you can see the rough ER there, you can see little membrane sacs going to the Golgi apparatus and out to the outside of the cell. So here is a little bit about proteins and where they might end up. If they are synthesized or translated by a ribosome free-floating in the cytoplasm, they're going to finish dissolved in the cytoplasm. But if they are made at the surface of the rough ER, they have a little, entrance sequence, and they enter, and they stay separate from the cytoplasm for the rest of their journey through the endomembrane system. So once they're inside the ER, they can move around inside this tunnel-like system, that anthill-like system, and they can have all kinds of things done to them. Chemical modifications to change their shape and thus function can be done here. Disulfide bridges can be added. A lot of chemical modifications are done. And they can also be marked for a certain destination, since this is really a shipping system through the cell. Any proteins needed by the cell are going to be made in that free-floating manner by the free-floating ribosomes. And they're going to stay a part of the cell. But if it needs to be shipped out of the cell, it has to go through the rough ER and be a part of the endomembrane system. Proteins are tagged using carbohydrates. So you can see here an example of a carbohydrate tag. These are generally oligosaccharides, so they're short polysaccharides, but they are longer than, say, a disaccharide. And they're just little chemical shipping labels to direct the rest of the endomembrane system as to how to handle that protein the rest of the way. So I think this is really cool. It's like a little address label that's stuck onto the protein, and it goes with it as it travels through the endomembrane system. The smooth ER extends a little bit beyond the nucleus. So at this point, the ER is much more like a network of tubes instead of flattened sacs. There are no ribosomes here because the proteins are already assembled. But you can further modify the proteins here, more tags or more changes to their 3D structure. That's what I mean when I'm talking about modification. The smooth ER also is like an amazing chemical factory. It can hydrolyze glycogen. It can change the chemical composition of harmful substances, making it very, very handy for, say, liver cells to have since they are detoxifying the body. And it can also synthesize lipids. Lipids, again, we've talked about this before, but they are the primary components of membranes, both the outer plasma membrane and all of these membrane-enclosed organelles we've been talking about. Because the lipids are hydrophobic, or at least they have the long hydrophobic fatty acid tails, they are going to remain separate from the rest of the cell. That means that tiny bubbles or sacs of membrane can carry things from one place to another by budding off of one larger membrane, traveling through the cytoplasm, and fusing into another large membrane. It's going to empty its contents into the place where it fuses, and that's exactly what vesicles do. So vesicles are just tiny sacs of lipids that travel. They're like little vehicles. They're going to carry things, their chemical cargo, those proteins that they're carrying, from the ER to the Golgi apparatus, and from the Golgi apparatus to the cell membrane. And so here is what I'm talking about. You have the ER, you have the Golgi apparatus, and you have a little vesicle bud off and travel and then fuse in with the Golgi apparatus. So seeing that again in reverse, it buds off, travels across the membrane, fuses, and enters the Golgi. Because those lipids are able to stick to each other and remain separate from everything else, they can do this. And so those proteins never interact with the cytosol, with that liquid part of the cytoplasm. They remain separate the entire time. The Golgi apparatus is the next major stop on our endomembrane system journey. In vertebrates like us, the Golgi is arranged as a series of flattened discs. They're stacked on top of each other, very much like a short stack of pancakes or a bunch of plates in a cabinet. Vesicles enter the Golgi on the side closest to the nucleus, and that side is called the cis face. And then the vesicles bud off and exit the Golgi closest to the plasma membrane, which we call the transface of the Golgi. So what happens to the proteins as they travel through the Golgi layers? Well, that's going to be things like more chemical modification and a whole lot of sorting, gathering, and packaging. It's as if a whole lot of unsorted mail is dumped in on the cis side, and then as it travels through these layers and goes through the sorting process, it's able to leave in nicely organized packages, grouped by purpose or by destination, off the trans side of the Golgi apparatus. These organelles fuse with the plasma membrane, so those outgoing vesicles fuse with the plasma membrane and empty their contents outside the cell. So here is an example of that. And I want to point out while you're looking at this, you can see step 1, step 2, and step 3 here of how it would be emptied outside the cell. The gentleman involved with finding these docking proteins involved in this process won the Nobel Prize in 2013. I believe it was for physiology and medicine. So cell biologists looking at vesicle movement are winning Nobel Prizes, which I just love. Vesicles are they're very present all over the cell, and I feel like they can be easily forgotten about. But they are very, very important. Their job, so simple, moving things around, yet so important. Another thing I want to note while you're looking here is that if there were proteins embedded in the membrane of that vesicle, they could travel and become a part of the cell membrane. And so one thing the Golgi apparatus can do is make sure that proteins that need to be a part of the cell membrane are embedded into the cell membranes or the plasma membranes of the vesicles before they travel to the outer membrane. Pretty cool. And that brings us to what is most certainly my favorite organelle. In my early years of teaching, this poor organelle was often the most forgotten about, but hopefully you're not going to forget it now, because I'm going to sing its praises and I'm going to tell you all about it, and I've told you it's my favorite, and that alone gives you a good reason to remember it. The organelle looks just like a vesicle, and it buds off the Golgi's transface just like a vesicle, but we give it a different name because it has a different job, and we call it a lysosome. It's full of hydrolyzing enzymes, meaning it can go through hydrolysis and break stuff up, and it does not exit the cell. The lysosome is met by another vesicle. Often this vesicle is formed from a depression in the cell membrane, and it allows the cell to eat some of the material outside the cell. Often this includes large macromolecules that the cell is going to need to break down as food. So the early or primary lysosome, which is what we call it when it is just formed from the Golgi, fuses with what we call a phagosome, and it can start breaking down all those macromolecules with all of its hydrolytic reactions. And so that's digestion. So all of these enzymes are breaking down so many bonds. This is like a ferocious chemical feeding frenzy to me. And when the frenzy is over, all the tiny monomers now can diffuse through the membrane of the lysosome, and the lysosome can dump any undigested material by fusing with the plasma membrane and just dumping those things outside the cell. Lysosomes can do the same thing, and this is when they get really cool, with any kind of worn out organelles or any invader that had been captured, say, by a white blood cell. So they can feed, they can recycle, they can defend the cell. They are very amazing, adaptable little organelles, and it's all due to the hydrolyzing enzymes they contain. And those enzymes started out as proteins as a part of the endomembrane system, which wraps up this part of the cell lesson.